My name is Sadhana, and today I'll be presenting my work on unfolding the medial temporal lobe to characterize neurodegeneration due to Alzheimer's disease using ex vivo imaging. Alzheimer's disease is the most common cause of dementia amongst older adults and affects the lives of more than 6 million Americans. While the cognitive symptoms of AD are generally well known, changes in the brain actually occur 10 to 15 years before symptoms are even observed. There are 3 pathological hallmarks of Alzheimer's disease: amyloid plaques, which is the extracellular deposits of beta amyloid; neurofibrillary tangles, which is the intracellular accumulation of tau protein; and neurodegeneration. Studies have shown that the accumulation of NFTs in particular is strongly correlated with neurodegeneration. The medial temporal lobe is the part of the brain that's damaged the earliest and most severely in Alzheimer's. The MTL can be divided into several subregions. Here I've shown the hippocampus, the internal cortex, and Broadband area 35. The hippocampus has been very well studied in AD. However, the earliest manifestations of tau or NFT pathology are seen around Broadband area 35 and the lateral end of the entorhinal cortex. This accumulation of tau pathology is associated with volumetric changes, which can be measured using in vivo structural MRI using image analysis techniques, and measures of the structural changes are useful biomarkers for AD. The sensitivity of biomarkers can be improved by obtaining structural measurements from the medial temporal lobe, since it's the earliest region affected by NFT pathology. This figure shows the Brock stages, which describes the characteristic pattern of spread of NFT pathology within the MTL. As mentioned previously, early accumulation is observed near BA35 and the entorhinal cortex. The pathology then gradually spreads till it covers the entire MTL region before spreading to other parts of the brain. When trying to obtain granular measurements from the MTL, one of the challenges is that multiple other pathologies are frequently comorbid in patients with AD. These pathologies also affect MTL structure. However, it is thought that the different subregions are differentially vulnerable to the different pathologies. Therefore, by obtaining measurements of the MTL in regions which are affected specifically by AD, we can obtain better biomarkers that are more sensitive and specific to Alzheimer's. In our work, we aim to use ex vivo imaging, combining high-resolution MRI with 3D serial histology to help inform the development of in vivo MRI biomarkers. Ex vivo imaging combined with serial histology provides a direct link between MRI measures of MTL structure measured using MRI and the underlying pathology. We hypothesized that in vivo MRI biomarkers defined over MTL regions where structural changes correlate most specifically with AD pathology would be more sensitive to disease progression during preclinical AD. This slide provides an overview of our ex vivo imaging pipeline. We are acquiring autopsy specimens from a large number of donors for ex vivo imaging. These specimens are scanned at 9.4 Tesla with a resolution of 200 microns isotropic. Following scanning, we print 3D custom molds for each specimen, which includes prescribed cutting guides for cutting the tissue blocks. Using this approach, when sectioning, we know where exactly in MRI space each section is located, which makes the alignment of MRI and histology more accurate and less labor-intensive. Following sectioning, the tissue is stained for cytoarchitecture and tau immunohistochemistry and then digitally scanned. The scanned sections are then reconstructed into a 3D volume and aligned to MRI space using a custom deformable 3D registration pipeline. One of the objectives of this work is to label the subregions in the MTL based on cytoarchitectural boundaries defined in histology. Our collaborators at the University of Castilla-La Mancha in Spain marked the boundary annotation between MTL subregions directly on the histology images. Once the histology and MRI scans are aligned, as shown in these images, the MTL subregions are then manually traced in MRI space using the boundary annotation from histology. The figure on the right shows an example 3D reconstruction of the final histology-based subregion segmentation. Additionally, using the scanned tau immunohistochemistry sections, we have developed a method to generate heat maps quantifying the severity of tau burden in a histology image using a weakly supervised deep learning technique. The video on the right shows a heat map quantifying tau co-registered and overlaid on the corresponding MRI scan. The automated NFT burden measures generated by the network were shown to be consistent with manual NFT counts and semi-quantitative ratings of NFT severity provided by an expert neuropathologist. Given all of this data, we are interested in understanding the association between neurodegeneration in the MTL measured using MRI and tau pathology. For this study, I used data from 18 MTL specimens. For 11 specimens, I had histology-guided MTL subregion segmentations, and in 15 specimens, I had 3D quantitative maps of NFT burden. An important first step is to create a common reference base across all the subjects that can be used to perform statistical analyses. This reference base can then be used to characterize anatomical variability in the MTL examine correlations between MRI measures of structural change and the underlying pathology, and studied 3D distribution of NFT pathology. One of the biggest challenges when creating this normalized reference space, however, is that there is significant anatomical variability in terms of the shape and folding patterns of the MTL across different subjects. The different folding patterns have also been shown to affect the location of the boundaries of MTL subregions such as BA35 and BA36. There are two approaches for aligning the MTL across specimens. Typically, the approach is to construct a template which captures the average shape of the MTL using a volumetric to formal registration pipeline. And a big part of my work is focused on this approach. The challenge with this method, though, is that it can result in the collapsing or squashing of different MTL morphologies across subjects. The other approach, which I'll be talking about today, is to unfold the cortex and align specimens based on distance along the cortical surface. The advantage of this approach is that it explicitly accounts for the complex folding patterns of the cortex. However, existing in vivo tools for cortical flattening aren't easily applicable to our ex vivo images. Therefore, in this work, I instead use a topological framework developed by de Crocker et al., which can be adapted to create a 2D unfolded representation of the MTL. To provide an overview of the method proposed by de Crocker et al., the first step is to generate segmentation to define the region of the MTL to unfold. Since manual segmentation of the MTL will be very labor-intensive in our ex vivo data, we used a semi-automated segmentation tool developed in my previous work to label the MTL in each of the MRI scans. I also semi-manually labeled the source and sink boundaries along each axis of the MTL, which are used to guide the unfolding process. Using these segmentations, I then solved Laplace's equations along 3 directions of the MTL, namely the anterior-posterior axis, the proximal-distal axis, which goes along the length of the cortex, and lastly, in the direction that goes from the inner white matter surface to the outer pupil surface. This results in a potential field in each direction, which increases smoothly from 0 to 1. The anterior, posterior, and proximal distal solutions together form a 2D coordinate system, which can be used to map image features such as tau burden, thickness, curvature, and subregion label from 3D image space to a flattened unfolded space. This figure shows the unfolded representations of the different MTL features for an example subject. So far, I've applied this framework to 18 specimens, looking at thickness and curvature along the MTL surface. For 15 specimens, we have flattened maps showing quantitative NFT burden, and 11 specimens, we have the MTL subregion segmentations. Note that since there's a discontinuity in the segmentation for the extent of the MTL in the anterior and posterior region of the MTL, the unfolded maps are generated separately for each region. This figure shows the average thickness and NFT burden map computed across 15 specimens. Additionally, using the subregion segmentations available in 11 subjects, I was able to generate a consensus MTL subregion segmentation in this flattened space. Since only a subset of specimens have a subregion segmentation, I used this consensus segmentation to define a BA35 region of interest in each of the 15 specimens and computed a summary measure of NFT severity defined as the 90th percentile of NFT burden across all points of fall within BA35 in the unfolded maps. We used BA35 as a reference since it's the earliest region affected by NFT pathology. In this flattened space, I then looked at the pointwise correlation between thickness and NFT burden, and we observed significant correlations in the entorhinal cortex consistent with the early Brock regions and also parts of BA36. In an exploratory analysis, we were interested in looking at whether NFT distribution is dependent on distance along the cortex or a consequence of MTL morphology. Mean curvature can be used to align specimens based on sulcal patterns, since the fundus of the collateral sulcus can be observed as a region of high curvature in these maps. This is analogous to free surface in vivo surface-based registration method. Smooth groupwise intensity-based registration was used to align curvature maps across subjects. We then repeated the pointwise thickness analyses following sulcus-based registration and found no significant correlation between thickness and NFT pathology. This result is consistent with the results obtained when we performed a similar analyses in the space of the volumetric template and suggests that patterns of tau distribution and neurodegeneration may be more dependent on distance along the cortex. In conclusion, ex vivo imaging can be used to understand anatomical variation, directly correlate MTL structural change with the underlying pathology, and find regional hotspots where AD pathology correlates most strongly with MTL structure. The unfolding framework provides a valuable tool for characterizing neurodegeneration in the MTL. And in future work, my work will focus on increasing the size of the data set, extending the unfolding framework to include the hippocampal subfields, and using the same framework to study a different set of imaging features, such as other common molecular pathologies. Lastly, I wanted to acknowledge the tissue donors and their families, as well as the large team of people behind this work who've put in a lot of effort into this project at the different stages of the pipeline. Thank you for the opportunity to present today.